Antigens, major histocompatibility complex, and innate immunity. We're going to start with an overview of the immune system. So previously I discussed how our host defenses, or our immune system, is divided into the innate and acquired immune system. Innate is nonspecific. It will basically defend against anything that's foreign. Acquired, or the adaptive immune response, is specific. So it specifically binds to and recognizes certain types of foreign substances, and it mediates an overall attack against that substance. The innate immune system is then divided further into the first and second lines of defense. The first line of defense would include all of the nonspecific surface protection that we have that will prevent microorganisms from getting into the body. This includes physical barriers, microbiota barrier, and chemical barriers. Physical barriers is talking about physical, like skin cells that are in the way of microorganisms from getting in. Microbiota barrier is talking about the normal flora or microbiota that lives on the surface of our body and that protects us from getting certain types of infections. And you have to remember that the surface of the body is going to be not only the surface of our skin, but any external facing internal organs, which sounds kind of weird. But if you think about the inside of your digestive tract, when you swallow food, that food that's coming into your digestive tract is staying in a tube. And that tube is open to the outside environment. So the esophagus from the top is open from your mouth, and the large intestine is open from the anus. So technically, the inside of that digestive tract tube is considered the outside of the body. Just like if you think about the lungs. So the respiratory tract, the air comes in, it goes down the trachea, and then from the trachea, it's going to branch to each lung. The air circulates, and then the air comes back out the same way. But if you think about that pathway, air is never really entering into the body until it gets into our bloodstream. So we consider the inside of the respiratory tract the outside of the body as well. Similar as the urinary tract, the lower part of the urinary tract, when we go up from the ureter into the urinary bladder. The urethra into the urinary bladder. From the outside of the body, it's going into that bladder. We consider all of that component, all of that surface area, the outside of the body. Same thing as the vagina. The inside of the vagina is kind of considered the outside of the body as well, because it's a infolding of tissues. It's not actually completely enclosed inside the body. So when we talk about the microbiota barrier, we're not just talking about the bacteria that live on the surface of the skin. We can also refer to the bacteria that's inside of our respiratory tract, inside of our digestive tract, and inside the vagina, inside the lower part of the urinary tract. Chemical barriers is going to be talking about the chemicals on the surface of our body that are going to prevent the growth of microorganisms. So low pH, high salt concentration, microorganisms don't really like living in that environment. Then we go on to the second line of defense. The second line of defense, like let's say a microorganism broke through those barriers, and now we have bacteria or microorganisms spilling into the body. This usually happens because of some type of damage, some type of cut in the body. But once the skin is penetrated by these microorganisms, we have this non-specific generalized attack ready to go so that it can clear that infection from the body. Those different components include phagocytosis, inflammation, fever, and different antimicrobial products that we'll discuss in detail as we go through this lecture today. The third line of defense is more specific and more involved. So we'll get into the third line of defense a little bit more once we get to the adaptive immune response. So to start with some definitions, an immunogen is a macromolecule that's capable of triggering an adaptive immune response. So sometimes the terms immunogen and antigen are used exchangeably. They are different things. So an immunogen will trigger an adaptive immune response. An antigen may or may not. So an immunogen is kind of a type of antigen, but we cannot say all antigens are immunogens, if that makes sense. So immunogens will likely trigger that response. Antigens may or may not trigger that response, but they're going to react with an antibody or sensitized T cell. An antigen I like to refer to as the cell's ID card. It's an external molecule on the surface of the cell that your immune system can either recognize as belonging, self, like it's part of yourself, or being foreign, non-self. If it's non-self, then your immune system is going to mount an attack against it. If it's self, it should not. Going on to major histocompatibility complex, so MHC, these are molecules that must combine with an immunogen before T cells are able to respond. So they basically hold up a chunk of that organism, microorganism, or a chunk of that foreign substance. They're going to take off a chunk of that organism that has the capability of stimulating an immune response. So that chunk is called an immunogen. And they're going to hold it up to your immune system. And in different situations, different things are going to happen. And we're going to talk a lot more about MHC when we get into adaptive immunity. Immunogenicity is the ability of an immunogen to stimulate host responses. This depends on the macromolecular size, so how big it is, the foreignness, how different it is from us, the chemical composition and molecular complexity, and the ability to be processed and presented with MHC molecules. An epitope is the key portion of the immunogen that's recognized by the immune system. It's the actual part of that immunogen molecule that the immune system will bind to and recognize as being foreign. Haptens are non-immunogenic materials that can combine with a carrier to create new antigenic determinants. For example, poison ivy contains catechols that combine with proteins in the skin to form immunogens that produce the reaction. Adjuvants are administered with an immunogen, typically in a vaccine, to increase the immune response. Adjuvants are going to stimulate or target antigen-presenting cells. Antigen-presenting cells are then going to mount a huge reaction in the presence of the adjuvant compared to if the adjuvant wasn't there. So it really just makes vaccines a bit more effective. It makes antigen-presenting cells more excited to see that vaccine immunogen coming in, and then it will mount a more significant controlled response so that we get a good amount of antibody protection later on. So we talked about what an antigen is, and there's different categories of antigens. Autoantigens are host or self-antigens. They should not evoke an immune response, but in certain immune disorders, such as autoimmune diseases, they may evoke an immune response. Alloantigens are from members of the same species, but could elicit an immune response. So if you think about like the blood cell antigens, they're all from the same species, but if we do a blood transfusion from a blood type A person to a blood type B person, we could see a major reaction. So that's where alloantigens would come into play. Heteroantigens are going to come from other species. So if we were to put something from another species into your body, those would be considered heteroantigens, and your immune system would respond to those. Heterophile antigens are closely related or identical antigens from unrelated plant or animal species. So coming back to this major histocompatibility complex, me personally, I associate MHC with the adaptive immune response, and particularly talking about how the cells of the adaptive immune response are stimulated, but the textbook kind of clumps MHC together within this chapter. So we are going to talk about it here, but once we talk about the adaptive immune response, this will come back and we'll talk more about this activation of those T cells. So MHC was originally called human leukocyte antigens, HLAs. HLA and MHC can be used interchangeably. you will see that even within my own assignments, sometimes I use them interchangeably. HLAs are more associated with, like more commonly used when we're talking about molecular, or if we're talking about looking at the genes associated with this, while MHC is more related to like the immune response. But either way, both of these terms are used interchangeably. So you should just know HLA, MHC, same thing. You should also know what these stand for though. So HLA is for human leukocyte antigens, and MHC is for major histocompatibility complex. We changed the name from HLA to MHC because it's related to whether transplanted tissue is histocompatible. Histocompatibility complex. So it determines whether we reject it or accept it. So if a person gets a transplant, we have these molecules that will interact with that transplanted tissue. And if they have completely different molecules, our immune system may recognize that as foreign and may attack it. These are found on all nucleated cells. Nucleated cells means that they have a nucleus. The major cell in the body that does not have a nucleus are going to be your red blood cells. So red blood cells don't have these, but all nucleated cells in the body will. The main function is to bring antigens to the cell surface for recognition. MHC or HLA genes are highly polymorphic. This allows for the immune response against a diverse number of immunogens. They're located on the short arm of chromosome 6, and there's class 1, 2, and 3 HLA genes. Class 1 and 2 genes code for molecules that are involved in antigen recognition. Class 3 genes code for select complement proteins and cytokines. These are secreted proteins that have an immune function but are not expressed on cell surfaces. So rather, they're secreted into the bloodstream and they stimulate other cells. There are numerous alleles or versions of the genes that are possible. MHC class 1. MHC class 1 is located on every nucleated cell. Higher quantities are found on lymphocytes and myeloid cells, and there's low levels on liver hepatocytes or liver cells, neural cells, muscle cells, and sperm. This is why HLA matching is not done on liver transplants. The main role is antigen presentation to CD8 cytotoxic T cells. So this is a picture of the cell membrane, and then here is class 1 molecule. The role of class 1. So MHC class 1 is found, like I said, on all nucleated cells. So what this does is if a virus happens to get inside of a cell, let's talk about like the cells of your throat. If a virus happens to get inside of the cells of your throat, the virus's goal is to divide and make many copies of itself. But your cells don't want to be virus-producing factories. So what your cells are going to do is when that virus is inside, your cell is going to very carefully attach a component of the virus, attach an immunogenic component, an immunogen part of the virus, to a special arm called MHC class 1. And when it does this, it's going to pull that up to the surface of the cell, and it's going to hold that up to your immune system. And it's going to say, hey guys, there's this thing inside of me. I'm now a virus factory. You should come kill me. So then your immune system cells are going to be floating around inside of your throat, and they're going to see that antigen or that immunogen that's presented on that arm. And they're going to say, whoa, we've got MHC class 1 holding up a molecule. That means that this cell is infected. We have to kill it. So there's certain cells called CD8 positive cytotoxic T cells. They're going to bind through a series of reactions, and their goal is to kill virally infected cells. They can also do this with select other types of cells, cells that are infected with different types of intracellular pathogens. They're just like abnormal cells, but for the most part, it's going to be those virally infected cells that are going to hold this molecule up on MHC class 1, and then we kill those cells. Now, if we go back and we look at this, they're not going to be found on liver cells, nerve cells, muscle cells, and sperm, because typically we don't want to kill those cells. Can you imagine if your brain got infected with a virus and it had MHC? Well, then your immune system would start killing off all your brain cells, and that would be terrible. It would be devastating. So we don't want that to happen. Same thing with muscle cells. Like muscle cells, they aren't regenerated. You are born with all the muscle cells that you have. They can grow larger or smaller, but you typically don't like replicate your muscle cells. Hepatocytes can be regenerated at really fast rates, and they multiply all the time. But we don't want to kill off our liver cells when we get infected with certain viruses. So those types of cells don't have these types of molecules in high concentrations. But if you think about epithelial cells, or the cells that line the inside of your throat or your skin, we've got abundant supply of those. So if we kill one, it's not going to be a big deal. The benefit of our immune system killing it is at the moment that it kills it, that cell is no longer producing more viruses. One of the bad things, though, is when it kills the cell, it releases any viral particles that were produced. So sometimes when you start to mount an immune response, you get sicker and you start to feel crummier before you get better. And that's because the immune cells start killing those virally infected cells, releasing more virus quicker, and it causes just a lot of inflammation and upset in the area. And then it takes some time before all those viral particles can get released or get eaten by phagocytosis. So now going on to MHC class 2. So MHC class 2 is located predominantly on your antigen presenting cells such as B cells, monocytes, macrophages, dendritic cells, and thymic epithelium. Thymic is referring to the thymus. That's that big organ in your chest that shrinks as you age. This is where T cells develop, and the reason why it shrinks while you age is because we'll talk a lot more about this when we get to adaptive immunity, but children need to develop their whole arsenal of different types of T cells, and you stop producing new types of T cells as you age. That's why elderly people are much more immunocompromised than young people, because by the time you get to the end of your lifespan, a lot of those cells have died and you're not really producing a lot of new ones at that point. The main role for MHC class 2 is antigen presentation to CD4 helper T cells. So at MHC class 2, you got these antigen presenting cells. They're gobblers. They're going to go and eat foreign organisms that they find in the body. And when they eat them, they're going to chop off part of them and hold them up to your immune system on that MHC class 2 arm. Then CD4 from the helper T cell will bind to MHC class 2, and that T cell will then get activated to go activate other cells in the area. So one thing that you should make note of, and this will be a quiz question and possibly a test question, is the association of CD4 with MHC class 2 and CD8 with MHC class 1. You should also know that the CD8 positive cells are called cytotoxic T cells. So a CD8 positive cytotoxic T cell would bind to MHC class 1. A CD4 helper T cell would bind to MHC class 2. So you can go ahead and pause the video if you need to and make note of that. That's an important point. So what's the clinical significance of MHC or HLA? Testing for MHC before tissue transplant, transitioning from serological to molecular. So we used to just mix some cells together and see what happened in the reaction before we did an organ or tissue transplant. Now we're actually doing some genetic or molecular evaluation. So what we'll do is we will do the DNA, I think that's on the next slide. Here it is. So we will do a DNA extraction. Then we're going to perform polymerase chain reaction, which makes a lot of copies of the HLA genes. And then we're going to sequence those genes, and we match the sequence of the patient to known sequences of various genes to figure out which genes are actually present. And then we can see if those would be compatible so that when we do this transplant, we're not going to have major rejection of the organ. MHC antigens play a role in the development of autoimmune diseases. So certain HLA diseases or genes predispose a person to certain autoimmune diseases. For example, HLA-B27 is associated with ankylosing spondylitis, which impacts the vertebrae of the spine. So in addition to worrying about it for transplant, we also worry about HLAs for select diseases. So now switching gears to talk a bit more about innate immune responses. So we've got the first and second lines of defense. The first line of defense is our external defense system. The second line of defense is our internal or nonspecific defense system. It includes pathogen recognition receptors, acute phase reactants, inflammation, phagocytosis, and natural killer cells. So when I teach this for my general microbiology courses, I typically give the scenario of you bumping into someone on the street. So if you bump into someone on the street and most of the time you say sorry and you go about your way, that's kind of like when a microorganism comes in contact with the surface of your skin. The surface of your skin has a whole bunch of barriers and chemicals that will prevent any type of penetration or interaction. So let's say that when you bumped into that person, you say sorry, and they're like, no, apology not accepted. We are going to take it to the next step and start a fight. And you do everything that you can to prevent the fight. And you're like, no, let's not fight. But this person is adamant on fighting. Then you have to get into your second line of defense. So if that microorganism happens to penetrate those external barriers, let's say you have a cut on the surface of your skin, or the microorganism is specifically designed to thrive on the skin, then it could invade into the tissues a little bit deeper. And once it's in your tissues, now it can cause a secondary type of, or a deeper infection, which relies on our second line of defense. So now it's inside the body. We need our internal defense system. So you by yourself on the street, you're like, okay, this person wants to fight me. So you pull out like your nunchucks and you're ready to fight this person. And you're doing everything you can. You kick them and punch them and fight them. That's kind of what our non-specific defense is. So it's going to try to do all of these little things in order to clear that pathogen from our body. Eventually, after you get to a certain point, you're either going to lose the fight or you're going to have to call for backup. So maybe you get on the phone and like you call your cousins and you need to bring your cousins into the fight. So when you call for backup, that's calling for your third line of defense. So we're going to stop kind of this scenario here because the third line of defense goes with the adaptive immune response, and we'll be talking about that later on in the course. We're just going to focus on what your body can do for that second line of defense when the pathogen actually gets past the physical or the external barriers, and now it's in your body causing an even bigger problem. So here's that external defense system. You can see cough and sneeze reflexes expel microbes from the body. Lacrimal glands are your tear glands. Salivary glands for saliva. Those contain lysozyme, so there's antibacterial properties to lysozyme in your tears and saliva. Airways, and back to the saliva and the lysozymes. So if you've ever wondered how like coyotes could eat a rotten carcass on the side of the road and not get sick, but if you were to do the same thing, you'd get violently ill. It has to do with the types of enzymes that they have in their saliva to be able to kill off those microorganisms and survive when coming in contact with those pathogens. We don't have that many enzymes in our saliva, so us eating something that's rotten flesh would definitely kill us as opposed to those animals that can do that. But that's just a side note there. So airways has mucus and cilia. So the cilia expels mucus. Cilia are those hair-like projections that are going to brush mucus out. And the mucus is going to trap the microbes. Skin has keratinocytes. These are a physical barrier. So the keratinocytes are basically dead pouches of keratin. So everything that you see on the outer surface of the cell is dead on a person, except for the cornea of the eye. That is the only like living tissue you can see when you're looking at a person standing there with their mouth shut. So that's kind of interesting that everyone sees dead people, but everything on the outside surface of the body is considered dead. Sweat glands contain lactic acid, sebaceous glands contain fatty acids, and then you have the low pH and high salt concentration of the skin that prevents the growth of a lot of different microorganisms. The stomach contains gastric acid, which deters the growth of a lot of microorganisms. But there's certain species of bacteria that can live and survive through even a pH of 1 or 2 that they encounter in your stomach. Helicobacter pylori, as an example, is one of those organisms that can live in the presence of acid in your stomach. That one has the capability of causing ulcers, so gastric ulcers in the stomach. So just kind of summarizing what I've been talking about here. So skin, physical barrier with secretions that discourage microorganism growth, low pH, high salt. Low pH, also known as a acidic pH. So high pH is basic, low pH is acidic. Serracin is a small protein produced by skin cells that has antibacterial effects. The epidermis is tightly packed epithelial cells that are coated in keratin. They're actually sacs of keratin. And the dermis has connective tissue with blood vessels, hair follicles, sebaceous glands, sweat glands, and white blood cells. The respiratory tract has those mucus secretions, and then, like I said, coughing and sneezing moves pathogens out of the tract. Urine helps flush out pathogens from getting up the GI tract. And the digestive tract, the stomach's hydrochloric acid, prohibits microbial growth, and then you have the normal flora that helps prevent pathogens from establishing themselves in those areas. So if it does get past all that, and it's able to start colonizing the body and infecting the body, we have that internal defense system, which consists of pathogen recognition receptors, acute phase reactants, inflammation, phagocytosis, and natural killer cells. Pathogen recognition receptors, PRRs. If you just pause and think about those terms, because sometimes when terms are thrown, even at me, I'm like, wow, that's a big term, and it's like hard to digest. Pathogen recognition receptors. What do you think those can do? They're receptors that recognize pathogens. Where do you think those would be good to be on? Probably some type of white blood cell. So PRRs are in fact found on macrophages, dendritic cells, neutrophils, eosinophils, mast cells, T cells, and epithelial cells. They help distinguish self from non-self using pathogen-associated molecular patterns, or PAMPs, that are only found in microorganisms. So PAMPs are on the surface of microorganisms. So if an organism comes into the body and it has a PAMP, we know it's foreign. It's not self because it's something that's on a microorganism. If it doesn't have a PAMP, maybe it does belong in our body. But once a PAMP binds to, or once the PRR binds to a pathogen and it activates phagocytic cells, it causes a rapid production of cytokines and chemokines. These are chemical messengers that we'll learn about later on in this course. And those chemical messengers are going to send signals to other immune cells to get cells active. They can also influence other parts of the body, like causing vasodilation, or the blood vessels to expand. Some cause vasoconstriction. The blood cells or vessels will constrict. So different things in the body can be controlled by these different types of chemicals. Toll-like receptors, or TLRs, are a major type of PRR. So PRRs, pathogen recognition receptors, are a big, huge category. One of the things within this category is called a toll-like receptor. TLR 1 through 10 have been identified in humans, and here's a table with the different targets and what is actually recognized. You'll notice with TLR 10, we don't know what it does yet, so be on the lookout for research on that one. Acute phase reactants are soluble factors that are found in serum. They increase rapidly in response to infection, injury, or tissue trauma. If you're taking clinical chemistry or if you've taken it in the past, you know that there's different chemicals that you can test for in the blood, and higher levels of certain chemicals can indicate certain disease states. While high levels of acute phase reactants typically will indicate some type of infection, injury, or tissue trauma. These acute phase reactants are going to facilitate contact between microbes and phagocytic cells. So they're going to help phagocytic cells find and consume microbes faster. They're also going to mop up and recycle important proteins after phagocytosis. So this is a table of some of the common APRs. The most common one that is tested for in the blood and is associated with inflammation is going to be this C-reactive protein. So if you see high levels of C-reactive protein, sometimes that means that yes, there's inflammation going on. Sometimes it's not inflammation. But if we see elevated levels and we know that the patient has an infection, then it kind of makes sense to laboratory scientists. The inflammatory response. So the inflammatory response is the body's overall reaction to injury or invasion by an infectious agent. It's the body's response to tissue damage. You can have inflammation even if microorganisms don't get in the body. Like let's say you roll your ankle, you're tearing tissue inside of your ankle. It doesn't tear open from the outside of the body. It's not like microorganisms are rushing in there. It's just injury. So you could have injury, like tissue damage, or you could have an invasion of infectious agents, or both. And that's going to stimulate the inflammatory response, or inflammation. There are 4 cardinal signs of inflammation: redness, heat, swelling, and pain. So the first thing that happens is the resident macrophages and mast cells at the site of infection or injury are going to release chemokines that cause vasodilation and induce selectins. So macrophages in the area are going to sense that there was some tissue damage, and mast cells are going to sense that there was some tissue damage, and they're going to start releasing chemicals, and the chemicals are going to cause the blood vessels to dilate. This is going to allow nutrients to leak out into the tissues to get nutrients to where they're needed in order to rebuild those tissues. But it's also going to allow for neutrophils to be called to the area. So circulating leukocytes called neutrophils, those guys are going to be stimulated, and they're going to follow that chemical signal all the way back to the source of the injury. These neutrophils are going to squeeze through your blood vessel walls, and then they're going to emerge and get to the site of infection or injury. All the while that this is happening, since your blood vessels are dilating, you're going to have that swelling happen in the area because fluids and cells are leaving your bloodstream to go to the area of damage. So you're going to have swelling, and swelling causes pressure to build up, which is painful. So that's going to stimulate your pain receptors. Since your blood vessels are dilating, that's also going to cause more blood to come to the area. Blood is typically hotter, like your body temperature, 98.6 degrees, typically hotter than the outside temperature. So you're going to see the redness because of the blood hue from underneath the skin, and it's going to feel hot because the blood circulating in that area, the increased blood in that area, is going to heat up the surface of the skin. Selectins loosely bind circulating leukocytes and cause them to roll along the vascular wall. Chemokine-induced integrins on the leukocytes bind firmly to the endothelial cells, and integrins enable the leukocytes to crawl between endothelial cells, known as a process called diapedesis. So selectins are going to help it bind to the surface of this blood vessel, and eventually it's going to help it squeeze through the blood vessel wall. Then they're going to follow that chemical gradient back to the source of infection. Diapedesis is kind of squeezing and lurking forward. Ped refers to foot. Diapedesis, you can think of like an amoeba kind of oozing along. That's kind of what that refers to. And then we have chemotaxis. Taxis is movement, chemo is chemical. Chemotaxis refers to the movement in response to chemicals. There can be positive or negative chemotaxis. In this case, it's positive chemotaxis because the chemicals are calling something to move toward them. Negative chemotaxis would be in the instance when a chemical causes something to go away. But in this instance, we're moving something toward the site of inflammation, so that's a positive chemotaxis. Phagocytosis, so your primary eaters are going to be your neutrophils, monocytes, macrophages, and dendritic cells. You do not need to know all of the details here involved in phagocytosis, but basically the microorganism is going to be internalized. Once it's internalized, it gets into a thing called a phagosome. And the phagosome is a pocket that contains that microorganism. In the cytosol, you have these organelles, these membrane-bound organelles called lysosomes. Lyse means to break apart. Lysosomes contain digestive enzymes that are going to help break apart anything that it comes in contact with. So a lysosome is going to fuse with a phagosome. And once it's fused, these chemicals are going to start breaking apart the microorganism, and eventually it's going to release it by exocytosis. And then all of those particles that are released are then going to either be recycled or they're just going to be circulated out through the urinary system. Natural killer cells provides defense against cells that are virally infected, cells that are infected with other intracellular pathogens, or tumor cells. In other words, weird-looking or abnormal cells. If you got a virally infected cell, it takes on a different shape. It looks weird. If you have a cell that's infected with another type of intracellular pathogen, like chlamydia is an intracellular pathogen, the cell is going to look weird. Or if you have a tumor cell, that cell is going to look really weird because it's going to be made super fast and it's going to look unstable and weird shaped. Natural killer cells are going to recognize that, and they can eliminate it without prior exposure to it. That gives the immune system time to activate the adaptive immune response of specific T and B cells. It's stimulated by the exposure to cytokines. So when it's activated, it produces additional cytokines and colony stimulating factors. This influences innate and adaptive immunity. So here's a natural killer cell. Here's a target cell. If this target cell has certain MHC molecules on it, MHC class 2, this would not be activated, and we're not going to go ahead and kill that. Because if it does have certain molecules, those means that those types of cells are beneficial to us. But if it doesn't have those beneficial molecules, it's going to be activated. So virally infected target cells don't have those. Antibody-coated target cells don't have those. Anything like that it recognizes, it's then going to kill the infected cells, and it's going to cause those cells to go through apoptosis, or programmed cell death. So in summary, the immune system is separated into the first, second, and third lines of defense. HLA, MHC, plays a major role in mediating immune responses and disorders. The innate immune system reflects the first and second lines of defense and includes external and internal barriers. The adaptive immune system reflects the third line of defense. Internal barriers against infection include nonspecific binding of foreign substances to immune receptors and proteins, inflammation, phagocytosis, and natural killer cells.